Dear student, I am Dr. Prakash Mungli, professor in biochemistry. So in this video, I will explain you all about enzyme inhibition. What is an enzyme inhibitor? Enzyme inhibitor, it's an external substance or a chemical that has got a inhibitory effect on an enzyme. So broadly, enzyme inhibitors can be classified based on their action on the enzyme as reversible inhibitors and irreversible inhibitors. Now the reversible inhibitors, they are going to bind with an enzyme with non-covalent bonds, and recovery of enzyme activity is possible by dilution technique. That means when you increase the amount of substrate, so the effect of inhibitor can be decreased. Now, whereas, and what kind of inhibitors are there in a reversible category? It is a competitive inhibitor, non-competitive inhibitor, uncompetitive inhibitor, and feedback inhibition. Now, irreversible inhibitors they are going to bind with a covalent bonds with an enzyme. It's a tight bond formation between the inhibitor and a protein of an enzyme. So that means the recovery of enzyme activity is not ordinarily possible. So the type of irreversible inhibitors are non-competitive irreversible inhibitor, competitive irreversible inhibitor, and suicidal inhibitors. Now let's get into competitive reversible inhibition. Now the competitive reversible inhibitor, it's a structural analog, meaning that the structure or the shape of an inhibitor is almost similar to the shape of a natural substrate of an enzyme, as it is shown here in the figure. That substrate shape, it is very similar to the shape of inhibitor. That means there is a competition between the substrate and the inhibitor for the only one active site that is available over the enzyme. If a substrate binds to an active site, so there will be enzyme substrate complex formation, and that can make a product. If inhibitor binds to an active site by chance, then there will be enzyme inhibitor complex formation, and that means there is no product formation with that. So what is the effect of competitive inhibition on the kinetics of an enzyme? When you look at 𝐒𝐫𝐢𝐚𝐴 plot on the left hand side, so in the absence of inhibitor, look at the orange color line there. So where it will show a Km and Vmax of an enzyme. If you look at the blue line on the right side, so that is in the presence of competitive inhibitor. So Vmax remains same, but Km is increased. Why? Because in the presence of competitive inhibitor, so the natural substrate has got a hindrance here with the inhibitor. Inhibitor is trying to compete with the natural substrate for the active site. So in the presence of competitive inhibitor, substrate is not getting a chance here sufficiently to bind with the inhibitor. That's why what happens is, so it is taking time for a natural substrate to bind with an inhibitor. So in order to reach the same maximum velocity, we really need to increase the substrate concentration so that we kind of dilute the effect of competitive inhibitor with the natural substrate, so that natural substrate gets more chance to bind with the enzyme active site, thereby reaches the maximum velocity. So that means we are going to compromise by increasing the substrate concentration to reach maximum velocity here. So that means in the competitive inhibition, enzyme can still reach maximum velocity. Only thing is there should be increase in the substrate concentration that increases the Km of an enzyme. That means enzyme to reach half of its maximum velocity in the presence of competitive inhibition, it increases. So Km increases here in the presence of competitive inhibition, but Vmax remains same. These are the kinetics of competitive inhibition, that is Km of an enzyme increases, apparent Km increases, but Vmax remains same here. Now look at the Lineweaver-Burke plot on the other side, on the right hand side. In the Lineweaver-Burke plot, you are going to see both the lines here. they meet at the same point in the y-axis. So the y-axis, wherever the reaction velocity curve meets on the y-axis in the line-Weaver-Burk plot, it is indicative of 1 over Vmax. So that 1 over Vmax, it remains same in the presence of competitive inhibitor, but look at the x-axis interpolation there. So the point that meets on the x-axis in the presence of competitive inhibitor is nearer to 0 when it compared to where it meets on the x-axis in the absence of inhibitor. That means 1 over substrate concentration in the x-axis, it is going towards 0. That means there is an increase in the Km, because 1 over Vmax, it's a reciprocal number which is going towards 0 means value is increasing. So in the y-intercept, minus 1 over Km decreases, that means the Km is increased because it's a reciprocal number. So x-intercept minus 1 over Km decreases, so which is going towards the 0 means there is an increase in the Km, because the 1 over Km decreases value means Km is increased. So in the same way, like y-intercept 1 over Vmax, it remains same there. So make sure you remember and identify how to identify a competitive inhibitor both in the Michaelis-Menten plot and also in the line-Weaver-Burk plot. In the Michaelis-Menten plot, it is Km is increased, Vmax remains same, it is very clear there. Whereas in line-Weaver-Burk plot, you got to be careful. So in the competitive inhibition, so the y-intercept remains same, whereas the x-intercept minus 1 over Km is going towards the it is going towards the 0. That means minus 1 over Km decreases here. That means Km is increased. Okay. So line crosses in the competitive inhibition. C for C, competitive inhibition, line crosses, C for C, cross on the y-axis. Remember that. Now let's move on to see what are the examples for competitive inhibition. This table is giving you a list of competitive inhibitor and the natural substrate and the target enzyme. Malonate is going to compete with succinate for succinate dehydrogenase enzyme, which is there in the TCA cycle. Sulfonamide is going to compete with the para-aminobenzoic acid for thyroid synthetase, which is acting as an antimicrobial agent. Thyroid synthetase is used in the synthesis of folic acid in the bacteria. Dicumarol is a competitive inhibitor of vitamin K for epoxide reductase enzyme, and it is acting as an anticoagulant. Dicumarol is used as an anticoagulant. Methanol is going to compete with ethanol for alcohol dehydrogenase, and this happens in the methanol poisoning. And the methanol poisoning, so the treatment or antidote for methanol poisoning is ethanol, because when we give ethanol, which is a natural substrate, it's going to compete with methanol for alcohol dehydrogenase. Enalapril, captopril, they are going to compete with angiotensin 1 for angiotensin converting enzyme, and enalapril, captoprils are used in the treatment of hypertension. Now, neostigmine and edrofen, these are competitive inhibitors for acetylcholinesterase for the enzyme acetylcholinesterase here, and it is used, they are used in the treatment of myasthenia gravis. Statins are, they are going to compete with FMGE, that is hydroxymethyl glutaryl CoA for FMGE reductase enzyme in cholesterol biosynthesis. So statins are used as hypercholesterolemia drugs. And ethylene glycol, they are going to compete with the natural substrate ethanol for alcohol dehydrogenase, and ethylene glycol is used as antifreeze in locomotives. That's about competitive inhibition. Now let's move on to see non-competitive inhibition. In the non-competitive inhibitor here, this is not a structural analog. It's not a substrate analog. It do not resemble to the natural substrate. Non-competitive inhibitor do not resemble to the natural substrate, and also it is not going to compete with the natural substrate because it's not going to bind to the active site of an enzyme, as you see here. So the active site of an enzyme where the substrate is binding is shown, whereas the inhibitor site is different than the active site in a non-competitive inhibition mechanism. That means there is no competition here. So what happens with the non-competitive inhibitor here? So when non-competitive inhibitor, it is going to bind to the site other than the active site, which is an inhibitory site. So when it binds to an inhibitory site, what it does, it is going to change the conformation of an active site. So because the inhibitor binding to inhibitory site, so it leads to a conformational change in the active site, where it do not allow binding of the substrate to an active site. It doesn't fit properly, although substrate can bind to an active site, but it doesn't fit well. That means that additional bond formation generally do not go on, that the binding energy that has to be released to decrease the transition, means to decrease the free energy of activation to take substrate into transition state is not coming up. That means product formation decreases. Although substrate is allowed to bind to the active site even in the presence of inhibitor down there, but it doesn't allow proper fitting of the substrate because additional bond formation do not go on, and that overall decreases the conversion of substrate into product because the binding energy is not sufficiently coming. So because of this, what will happen? So the non-competitive inhibitor binding leads to conformational change in the active site of an enzyme that alters the reaction velocity, and the non-competitive inhibitor can bind either to the free enzyme or to the enzyme substrate complex, and inhibitor forms extremely tight covalent bonds with the functional groups, and thereby inhibitor binding site, thereby it is going to change the conformation of an active site there, and thereby non-competitive inhibitors are basically they irreversibly bind, and it is a permanent thing. It is not ordinarily removed. Non-competitive inhibitor not ordinarily removed by the inhibitor site, and thereby it effectively denatures the enzyme that they inhibit. So one more time, this non-competitive inhibitor is not a structural analog. So it is going to bind to a free enzyme, or it's going to bind to the enzyme substrate complex. By doing so, it's going to bind with an enzyme with a covalent bond, very tight bond, irreversible binding, permanent binding, and sometimes it may effectively lead to denaturation of an enzyme. So most of the non-competitive inhibitors are irreversible inhibitors. Remember that. So thereby, when the enzyme is totally denatured or when the enzyme is function is totally altered, not allowing proper fitting of substrate to an active site, so overall product formation decreases. So that means overall maximum velocity of the enzyme will decrease. Now let's see what are the kinetic changes in the presence of non-competitive inhibitors using Michaelis-Menten plot and Lineweaver-Burk plot. On the left hand side, you are seeing a Michaelis-Menten plot, and the line which is on the which is red in color, it is in the absence of non-competitive inhibitor, where you see it has reached a maximum velocity there, and also the Km is, you are seeing the Km there, which is point which is meeting on the x-axis. So now let's compare that with the blue line on the right hand side. Now look at the blue line. So although the Km in the presence, so blue line is in the presence of inhibitor, non-competitive inhibitor. So the Km is remaining same because it is the inhibitor is allowing substrate to bind with the active site, but that proper fitting is not going on, and the binding energy that has to be released sufficiently, and that's not coming up. Because of this, it is overall affecting the velocity of the reaction, and also enzyme can be denatured in the presence of non-competitive inhibitor. So overall velocity of the reaction decreases, and that's why you see that blue line where velocity is totally below the red line, indicating that there is decrease in the Vmax. So non-competitive inhibitor decreases Vmax, but Km remains same. Now look at Lineweaver-Burk plot. In the Lineweaver-Burk plot, so y-intercept, look at the y-intercept. So y-intercept in the presence of non-competitive inhibitor, it is going away from 0. So 1 over Vmax, y-intercept is 1 over Vmax, and that 1 over Vmax, it is going away from 0, indicating that Vmax is decreased, because the reciprocal number going away from 0, it is a decrease in the value. So whereas look at the x-intercept, that is minus 1 over Km, it remains same. So x-intercept minus 1 over Km remains same, means Km remains same, and y-intercept 1 over Vmax increased means So Vmax is decreased. So there is a decrease in the Vmax because 1 over Vmax, as it is going away from 0, so value is actually decreased because it's a reciprocal number. So Vmax, in fact, it is the decrease in the Vmax. So Km increases, Km remains same and Vmax is decreased in the presence of non-competitive inhibitor. Now let's move on to see some of the examples for non-competitive inhibitor. So usually heavy metal ions like arsenate they act as a non-competitive inhibitor. Example of enzymes where arsenate inhibits is glyceraldehyde 3-phosphate dehydrogenase and alpha ketoglutarate dehydrogenase complex, pyruvate dehydrogenase complex, alpha keto acid dehydrogenase complex. Now, iodoacetate can also act as a non-competitive inhibitor on glyceraldehyde 3-phosphate dehydrogenase enzyme. Fluoride acts as a non-competitive inhibitor on enolase enzyme, which is a glycolytic enzyme. Omeprazole acts as a non-competitive inhibitor on H plus K plus ATPase or the proton pump. Cyanide is a non-competitive inhibitor on cytochrome oxidase, which is present in the electron transport chain. Now, some of the other non-competitive inhibitor examples here: fluoroacetate. Fluoroacetate is a non-competitive inhibitor on aconitase. You know, aconitase, it's one of the enzyme in TCA cycle. Rat poison, fluoroacetate, inhibiting TCA cycle on aconitase. Heavy metal like lead, mercury, it has got an effect on ferrochelatase enzyme, and the ferrochelatase, it's an enzyme in heme synthesis. That's why lead and mercury can decrease heme synthesis, leading to anemia. Disulfiram, other name for disulfiram is antabuse, and this antabuse is a non-competitive inhibitor on aldehyde dehydrogenase enzyme, which is involved in alcohol or ethanol metabolism. Ethanol is converted to acetaldehyde done by alcohol dehydrogenase, and acetaldehyde is converted to acetate by aldehyde dehydrogenase. So disulfiram is going to inhibit the conversion of acetaldehyde into acetate by inhibiting aldehyde dehydrogenase non-competitively. Diisopropyl fluorophosphate, it is inhibiting serine proteases. Organophosphate poisons like malathion and parathion, it is a non-competitive inhibitor on acetylcholinesterase. Thereby, acetylcholine levels will be elevated in the neuronal junction, increasing the parasympathetic effects. Coprin is inhibitor of aldehyde dehydrogenase enzyme. So these are some of the examples for non-competitive inhibitor. Now let's compare between a competitive inhibitor which is reversible, competitive inhibitor which is irreversible, and non-competitive inhibitor which is irreversible type of inhibitor. Now, competitive reversible inhibitor is a structural analog. It's going to compete with the natural substrate. Competitive irreversible inhibitor is also a structural analog, whereas non-competitive inhibitor is not a structural analog. So there is whenever you see the term competitive, usually those inhibitors are structural analog because they are going to compete with the natural substrate. Now, competitive inhibitor, reversible competitive inhibitor can be overcome by increasing the substrate concentration by a dilution technique. Competitive irreversible inhibitor cannot be overcome by increasing the substrate concentration because it has bound with the active site of an enzyme irreversibly. Similarly, non-competitive inhibitor cannot be overcome by increasing the substrate concentration because non-competitive inhibitor, although it binds with irreversibly, but it is not going to compete with the substrate anyway. So there is no point in increasing the substrate concentration because it's not going to change anything. Coming to the binding to the active site. Competitive inhibitor, it binds to active site whether it is reversible or irreversible competitive inhibitor because they are going to compete for the active site with the substrate. Now the non-competitive, as it is there in the name, there is no competition. They are not going to compete for the active site with the natural substrate. Now effect on Vmax. Competitive inhibitor reversible, there is no change in the Vmax. Whereas competitive inhibitor irreversible competitive inhibitor decreases the Vmax because it's going to bind with an active site of an enzyme irreversibly. It is not coming out of that. So it is ultimately affecting the velocity of the reaction because that enzyme, once the competitive irreversible inhibitor binds there, that's the end of the enzyme action there. So Vmax decreases. It is very similar to a non-competitive inhibitor. So Vmax decreases. What is the effect of Km on all these three type of inhibition? Competitive inhibitor reversible, if Km is increased, whereas in the competitive inhibitor irreversible and non-competitive inhibitor Km is not changed. Pharmacodynamics, competitive inhibitor reversible decreases the potency, whereas competitive inhibitor irreversible and non-competitive inhibitor, they decrease the efficacy of the enzyme. So these are the differences between competitive inhibitor reversible, irreversible with non-competitive inhibitor. Now let's get into a little more into irreversible competitive inhibitor. So irreversible competitive inhibitor binds to active site irreversibly and brings an irreversible conformational change in the active site of an enzyme, and this leads to overall decrease in the available active site. Thus it decreases the Vmax of an enzyme. Km do not change because the binding affinity of an enzyme to the substrate before the inhibitor binding is not really affected. So only Vmax decreased, Km remains same like non-competitive inhibition. And the example for irreversible competitive inhibitor is phenoxybenzaimine, which is an irreversible competitive inhibitor of alpha receptors. It's a alpha adrenergic receptor inhibitor, phenoxybenzaimine. That's an example for irreversible competitive inhibitor. Now let's move on to see uncompetitive inhibition. Uncompetitive inhibition, as you see here, it is going to decrease the Km and also it is going to decrease the Vmax. As you see in Michaelis-Menten plot and also Lyon-Weaver-Burke plot. So in both the plots, you can see that in the presence of uncompetitive inhibition, there is a decrease in the Vmax and also they are going to decrease the Km. So Km is decreased and also Vmax decreased here. So both are decreased. So that's what is the uncompetitive inhibition. So in uncompetitive inhibitor won't resemble the substrate. It doesn't have any affinity for the free enzyme. So inhibitor binds to enzyme substrate complex. Thereby, it is going to decrease both Km and also Vmax. Example, inhibition of placental alkaline phosphatase by phenylalanine. Otherwise, other than this example, there is almost no other example that is encountered in medicine. Uncompetitive inhibitor decreasing both Km and Vmax. It is almost never encountered in medicine. Don't have examples for that other than placental alkaline phosphatase being inhibited by phenylalanine, which is an example for uncompetitive inhibition. Now let's move on to see another type of irreversible inhibition called suicide inhibition or mechanism based inactivation. So what is this suicide inhibition? It is a special type of irreversible inhibition where inhibitor are unreactive until they bind with the active site of a specific enzyme that it is inhibiting. So once the inhibitor binds to the enzyme, by the action of the same enzyme, the inhibitor is converted into its potent inhibitor form. It is converted into some other form which becomes more potent inhibitor and it's going to bind with the active site of an enzyme irreversibly, and that totally inhibits the enzyme action. And that's what happens. So that means, so the difference here is, let me give a write down here example. So we have hypoxanthine, which is normally converted into xanthine. Hypoxanthine is normally converted into xanthine, and that will be done by enzyme xanthine oxidase. XO is xanthine oxidase here. So there is a drug called allopurinol. So this allopurinol is a suicidal inhibitor of xanthine oxidase. So this xanthine oxidase can also act on allopurinol, and this allopurinol is catalyzed by xanthine oxidase. Similarly, how hypoxanthine was converted into xanthine, so allopurinol also converted into oxypurinol. So it is done by xanthine oxidase. So allopurinol is a drug. It's a suicidal inhibitor. It is going to go and bind with the xanthine oxidase, and xanthine oxidase is going to convert that allopurinol, which is looking similar to hypoxanthine. That's why xanthine oxidase is kind of confused. So it is going to consider that allopurinol as hypoxanthine, and it's going to take that allopurinol and convert that allopurinol into oxypurinol, and that oxypurinol, which is a more potent inhibitor of xanthine oxidase, what it does, it will irreversibly bind with the xanthine oxidase and do not allow the active site to be free. It is just totally occupying the active site and it's not coming out. So that means that's the end of the enzyme. Xanthine oxidase enzyme is done there. Okay, so that basically decreases the velocity of the reaction and totally alter the enzyme based reaction. So this type of inhibition, we call it as suicidal inhibition or mechanism based inhibition, because xanthine oxidase here, it is committing suicide by converting allopurinol into oxypurinol. Otherwise allopurinol would have not inhibited the enzyme. It is converting allopurinol into oxypurinol, and that oxypurinol is actually inhibiting xanthine oxidase there. It is like a backstabbing by allopurinol, because xanthine oxidase itself has converted allopurinol into oxypurinol, and oxypurinol totally irreversibly binding with the active site of this enzyme and inactivating the enzyme. That is what is a suicidal mechanism there. Example. So just now I have given you the example of allopurinol inhibiting xanthine oxidase. Other examples, we have trypanosomiasis treated by difluoromethyl-ornithine, which is inhibiting ornithine decarboxylase enzyme. And then we have aspirin, which acetylates the active site of cyclooxygenase enzyme, thereby inhibiting prostaglandin synthesis. So these are the examples of suicidal inhibition or the mechanism based inhibition. Coming to feedback inhibition or end product inhibition. This feedback inhibition or end product inhibition, it is not an external molecule that are inhibiting these enzymes. It is our own metabolic products which will have a feedback negative effect on our regulatory enzymes in the metabolic pathways. One of the example that I have given here in the cholesterol metabolism. So FMGE reductase enzyme, it is feedback inhibited by cholesterol, thereby decreasing the cholesterol synthesis. Whenever we have excess cholesterol in the cytoplasm, that excess cholesterol itself will have a negative effect on FMGE reductase enzyme, thereby decreasing the activity of FMGE reductase enzyme. So that's about different types of inhibition. So we have seen reversible and non-reversible, irreversible inhibition. In that mainly we have concentrated on competitive mechanism, non-competitive inhibition, uncompetitive inhibition, suicidal inhibition, also called as mechanism based inhibition, and the feedback inhibition. So that's all about enzyme inhibition. Thank you so much.